Welcome to Program Bachelor of Science 3rd year, the subject Botany, 5th semester, course code BOD 101, course title Plant Tissue Culture. This is Unit 5, Somatic Hybridization, and the name of this module is Cybrids and their Applications. I am Ms. Marina Janice Rodriguez, Assistant Professor in Carmel College of Arts, Science and Commerce for Women, Nuve, Goa. In this module, you will learn about cybridization, the objective of cybridization, methods to produce a cybrid and applications of cybrids. By the end of this module, you will be able to define a cybrid, recall the objective of cybridization, explain methods to produce a cybrid, and also state the applications of cybrids. Hybridization involves the fusion of protoplast of two different genomes. As we can see here, there are two cells, cell A and cell B, of two different genomes. They are phylogenetically different cells. The cell wall is eliminated either by mechanical isolation or enzymatic degradation, and as a result what we get is protoplast A and protoplast B. During fusion, these two protoplasts stick together like two soap bubbles. The plasma membrane fuses at small localized region at the point of contact, making a protoplast bridge. The bridge would extend and finally round off to form a fusion product. The types of fusion products obtained when the protoplast fuse are three types. One is a hybrid, which is having a combination of the nuclear genome as well as the cytoplasm. The second product is a heterokaryon, which has the presence of both the nuclei in the fusion of the cytoplasm. And the third one is a cybrid, which we are focusing in this module. The cybrid is having the presence of only one parental nucleus and the other parental nucleus is eliminated. As you can see in this figure, this is representing a cybrid. When the nuclei of one parent is eliminated, a cybrid is formed. So a cybrid or cytoplasmic hybrids, as we can call them, are eukaryotic cell lines which are produced by the fusion of two protoplasts from phylogenetically different species. The cybrids have a nuclear genome of one species, as the other nuclear genome is eliminated, and the cytoplasm of both the parental species. The objective of cybridization is to combine the cytoplasmic genes of one species with the nuclear and the cytoplasmic genes of another species. Cybridization provides unique opportunities for transfer of plasma genes of one species into the nuclear background of another species. It enables the recovery of recombinants between parental mitochondria or chloroplast DNA. It helps in the production of wide variety of parental or recombinant chloroplast with the parental or recombinant mitochondria. Mitochondria from one parental species may be combined with the chloroplast of another parental species. Production of a cybrid. First step is the protoplast isolation, wherein we eliminate the cell wall by means of mechanical or enzymatic method. Second, on obtaining protoplast after isolation, these protoplasts are induced for fusion through mechanical, chemical or electric method. Upon fusion, the protoplasts which have fused successfully are selected. This will be the selection of cybrid protoplast. After selection of cybrid protoplast, these are then cultured in a suitable culture medium. Upon development in the suitable culture medium, there will be regeneration of cell wall to form a cybrid cell. So at this point, the protoplast is changing from the protoplast stage to form a cybrid cell. As the development progresses, the cell will multiply to form callus. So there will be callus formation, and from callus formation there will be regeneration of plants. When the plants are regenerated, there is confirmation of cybridization by observing the expression for the desired character. Last of all, these plants, the cybrid plants, are multiplied. We know that during the production of a cybrid, the parental nucleus, one of the parental nucleus is eliminated. This is done by either irradiating the protoplast of one species with X-rays or gamma rays prior to fusion to inactivate their nuclei. Gamma fusion is used to bring about interspecific and intergeneric combinations, preparing enucleated protoplasts, that is the cytoplast of one species, and fusing them with a normal protoplast of another species. So these enucleated protoplasts can be produced by these ways, that is gradual elimination of chromosomes of one species of a hybrid cell during subsequent mitotic divisions. Elimination of nucleus of one species, or fusion of a normal protoplast of one species with an enucleated protoplast, or a protoplast with an inactive nucleus of another species. Applications of cybrids. The first one is means of genetic recombinations in asexual or sterile plants. Protoplasts of sexually sterile plants can be fused to produce fertile diploids and polyploids. This is a great boon for the agricultural industry. Overcoming barriers of sexual incompatibility. We know sexual incompatibility is a process wherein the pollen grain is rejected by certain stigma of plants, thus forming a barrier in plant breeding programs. So in plant breeding programs, sexual crossings at interspecific or intergeneric levels fail to produce hybrids because of sexual incompatibility. So the method of cybridization helps in this regard. It has found application in industry and agriculture. The third one is cytoplasmic male sterility. It is a phenomenon where the plant produces sterile pollen. Transfer of cytoplasmic male sterility along with other genes which code for the rate of photosynthesis, tolerance to temperature and resistance to disease of Cytoplasmic male sterility is a phenomenon where the plant produces pollens which are sterile. Through cybridization, there can be transfer of cytoplasmic male sterility along with genes which code for rate of photosynthesis, tolerance to temperature and resistance to diseases or herbicides. Experiments on cybridizations have resulted in plants with reconstructed cytoplasm combining mitochondrial DNA and chloroplast DNA encoded traits from both parents. To summarize, we have seen that cybrid is a cell with a nucleus of one parental species and the fusion of cytoplasm of both the parents. The fusion of protoplasts with the elimination of one parental nucleus forms a cybrid. Cybrids have various applications which include genetic recombinations, overcoming sexual incompatibility, and the transfer of cytoplasmic male sterility. These are my references. Thank you.